Okay, everybody, as promised, I am putting up an overview, my version of blood types and blood typing. I'm assuming if you're watching this that you have gone and read the portion in the book on blood typing that I assigned and the two other clips on YouTube that are not from me, but I'd like you to watch those before you watch this one. Another thing that might be a good idea is because there's some discussion about antigens and antibodies in this clip, you may want to go and watch some of the lecture clips. I hope you've been doing that a bit already. Clips 7, 8, 9 have to do with antigens and antibodies, and I think you'll understand a lot more about what I'm about to go over if you've seen some of that. All right, so without much ado, let's get going. All right. So the first thing I'd like everybody to realize is that what determines your blood type, I think it's actually question 8 in the questions I'm going to provide you, actually asks this question: what determines your blood type? And in one statement, I can tell you that your genotype determines your phenotype. And you might look at me and say, huh? This is stuff from 131. Genotype is your host of genes, right? Your DNA. The genes within that whole host of genetic material are coding for certain molecules, certain proteins. Proteins that will catalyze reactions that result in certain products that you're producing. So, in general, I can tell you that on one of the chromosomes where we have a gene for the A or the B antigen, and the other chromosome we have a gene that codes for an Rh antigen or a D antigen. These are what are determining your blood type. You could kind of answer this question in two ways. You could say, well, it's the genes that are coding for these surface markers, these antigens that exist on the surface of red blood cells. Or you could also kind of approach it and just say, well, it's the antigens on the red blood cells that determine your blood type. But I would like you to understand that back behind the scenes, it's the genes that are doing this. You're not going to change somebody's blood type easily, right? You would have to change their genes, okay? So we're talking about a location where we might have an A or a B antigen coded for, or we have another location where we might have an Rh factor or a D antigen coded for. And these antigens, these uniquely shaped molecules exist on the surface of red blood cells. All right. So, a little bit about antigens versus antibodies. Okay, and this will give us an opportunity to talk about the different blood types within the ABO blood type categories. If you're type A, we would automatically know that you have a gene in that location on the chromosome that results in an antigen that we call A. At least one gene. You could have inherited two genes. Of course, we have two sets of chromosomes, one from your mother, one from your parent. Or you could have just one gene, and the other gene is this recessive gene that doesn't code for anything, the O gene. Okay, so there are two ways to be type A, but you have to have at least one gene for A to be this way. Okay, what are the antigens you'd have if you are type A? A. Duh, right? By doing somebody's blood type, you would know they have that antigen. You wouldn't necessarily know how it is they're type A. You wouldn't know if they have one or two genes for it. Okay, to be type B, it's the same way. You're going to inherit a host of genes from each parent. You could have two B genes that you've inherited, or just one B gene and O gene from the other parent. Either way, you'd be type B and have B antigens on your cells. If you're type AB, well, in this case, you will have had to have inherited an A gene from one parent, a B gene from the other parent, doesn't matter which gave them to you, and you would have both A and B antigens on your cells. Finally, if you are type O, and this would be a recessive gene, okay? Sometimes because it is recessive, you will see lowercase i's used for this instead. But these two genes paired up together, giving you no antigen from either gene, would mean you have none. You don't have any of them when it comes to A's and B's. Okay? Now, the rule of thumb is when we talk about the antibodies you possess, if you're type A, well, it's always the opposite. Okay? You have antibodies to the opposite antigens. Okay? If you're type A, you're going to have antibodies to B. Type B, you're going to have antibodies to A. Type AB, well, you won't have any for these A's and B's. If you're type O, you're going to have both types of antibodies. Okay? All right, let's switch our attention to the D antigen, or the Rh factor. Okay? Again, we can talk about type, the blood type, the genes that make you that way, and the antigens you would possess. Okay? If you're Rh positive, this is a little simpler, okay? You either have the antigen and you're Rh positive, or you don't have the antigen and you're Rh negative. A little simpler. There's not like with the ABO, there's two different things that could code for an antigen there, and this other recessive one that doesn't do anything, right? Here, you either have a gene for it, or the gene doesn't make it. Okay? So if you're Rh positive, you could have, and we're going to use the D, because usually that's what we're talking about. The uppercase D would be dominant for making the antigen. Okay? So you have two genes here. Sometimes in my questions, I just use plus for this when I talk about this, because you have basically two genes that would code for the Rh antigen versus a gene that would code for it and one that would not. Either way, one gene for that antigen will result in the production of that antigen, and you would have the Rh factor. You are indeed Rh positive. Okay? Now, the only way to be Rh negative would be to have two genes, neither of which give the Rh factor, lowercase d, little lowercase letters here, okay, for being recessive, and you'd have none of these antigens. Now for this, when it comes to the antibodies you would possess, I wouldn't expect anybody who is Rh positive to make it an antibody against its own, that person's own Rh factor. So your immune system would know better, okay? Immune cells, B cells would never make antibodies against an Rh factor if you're positive. But if you're Rh negative, it can happen. It's not a guarantee though. It's not quite like it is with the A, B's, and O's, where you're automatically going to have the opposite antibody. In this case, a person will create antibodies against the Rh factor, anti-Rh or anti-D, we might say, if they are Rh negative. The person's Rh negative, so they're not going to produce antibodies against themselves, but they have to have been exposed first. Okay? So an Rh negative person can make antibodies against Rh, but they'll only do it if they're exposed first. Okay? An example would be something you've already read about and watched a clip about, and that is if you have an Rh negative mother, and that Rh negative mother has a child that's Rh positive. She will ultimately be exposed to the Rh factor and start producing these antibodies. And that you've already read and listened to a clip about. All right. Now, how is it that we can use the idea of antibodies as a handy tool in the laboratory, a diagnostic tool? Okay? If we have antibodies attacking our own red blood cells in our own bloodstream, this is bad news. We don't want any of that to happen. Any place where an antibody is bound to something in our body is flagged for destruction, right? Antibodies flag different things for destruction, and then phagocytes go and gobble them up, okay? So agglutination, this idea that you'd have all these antibodies binding up red blood cells in the bloodstream would be a bad deal. But we can use this to our advantage in a diagnostic lab. We could take somebody's blood, and let's just say I'll share with you behind the scenes I know this is B positive blood, okay? But let's say the person doing this test doesn't know that, okay? So what I usually do is I draw, okay? What is B positive blood going to look like for me? And I'm just going to use something that actually looks like a B here. So this looks like a B sticking off the cell. And I'm going to use a little square for an RH factor or my D factor, okay? If I add antibodies to A, to a bunch of cells that are in a drop of blood that have no A's on the cells at all, it's not going to have an effect whatsoever, right? It's not going to agglutinate, clump up these cells. So these cells are going to be out there like this. There's B's and RH factor on the cells. The antibodies are not going to bind to these, right? You have to imagine in here you might have an antibody like this, right? And it has a triangle shape out on the end. And these antibodies would not bind to anything we have on the cells because this person doesn't have A on their cells if they're B positive, right? So you would see just like you see here, anti-A was added to this second drop here, and you see no clumps whatsoever. It looks like it would just like the blood sample that is our control. Okay? Okay, so now let's consider the next drop where we add antibodies against B. Now, antibodies against B, I have to imagine these antibodies would have the shape that would stick to a B antigen, okay, something like this. And all antibodies have two sides to them, at least, okay? So what's this going to look like when I add antibodies against B? This is what's going to happen. They're going to agglutinate. They're going to stick to one another. And when they do, this is when we're going to see all of this business going on, all these little clumps that you can see with the naked eye. This tells us, aha, B is present. I now know that B is on these cells. So this person who goes in not knowing anything about the blood, this tells them a little clue. Okay? Then we have to do for one more antigen. Remember, the O gene doesn't give you anything, right? So you can't look for an O. There is no O protein, right? So we're done with the ABO system. We have to turn our attention to the RH factor, which is this D antigen. Okay, so next one, we're going to look for this particular, let's see here, we're going to look for something that might stick to the RH factor. So we're going to add antibodies against RH, or you could say anti-D if you like, and it's going to stick to those, right? If that's what happens, this is what we're going to see. We're going to have agglutination again. Again, we're going to look. We already made our decision. Okay, check mark. There are B antigens because it clumped up. Over here, no A antigens. Here we see agglutination. So, we're going to have B positive blood. That's what we see in front of us. Okay, you would want to be able to read something like this based on this kind of a result, be able to tell me, like on our quiz later on, right? Tell me what the blood type is based on some kind of result like this coming from a diagnostic lab. All right, let's talk a little bit more about blood typing and how it is that someone might be able to receive a transfusion and what types of blood could they receive. Okay, so first let's consider, we're going to do 3 examples. Let's do B positive blood. If I get a question or questions like this, what I would do is I would write down, and I'm going to allow a single blank piece of paper, front and back, for you to use on this next quiz, okay? Write down what you know. If a person has B positive blood, I want to draw a picture so that in my mind I can kind of envision what's going on. So B positive, I write down B positive. Immediately I write down what I know about someone who has B positive blood. If it has B positive blood, this means there's a B on their cells and there's a RH on their cells, just like previous examples we were just doing. So there it is, B's and RHs. I also know that this person would automatically have the opposite when it comes to the ABO system, when it comes to antibodies, right? So as for what antigens they possess, Bs and Rhs. What antibodies do they possess? Absolutely anti-A. Is there a possibility a person who is positive would have antibodies against the Rh, though? I would have to say no. Our immune system doesn't do that to us, right? If you have a positive blood, you have Rh factors. If everything's going right, your immune system would never make antibodies against that. So this is what we're dealing with. So now I have to answer the question, who can this person receive a transfusion from? And the answer is any blood that this person doesn't attack. So what we need to bring our attention to whenever we try to answer this kind of question is the antibodies they possess. If they have antibodies against A, I do not want to give them A's in any way, shape, or form, whether it's A blood or whether it's AB blood. And that's the key for this person, okay? I can't give them A, I can't give them AB, but there's two other kinds I can think about. I can give them B blood without the Rh factor, and it doesn't really matter for Rh here because this person will never make an antibody against it. I can give them O blood. O doesn't have any A's on it, so we're good. And there's no Rh's, but again, not really a factor because there's never going to be antibodies against it in this person's bloodstream. And because of that, I could also give them positive blood, right? So this person has 4 different blood types at their disposal that they could get if they were in need of transfusion. Let's do another one. Let's do the same questions, and we're dealing with O negative blood. Okay, what antigens do they possess? I want to draw myself a picture. It's O negative blood. I have to envision the blood cell. It's going to have no A's on it, no B's on it, and it's negative, so there's no Rh factors on it. There it is, bare naked cell, right? No A's, no B's, no Rh. Now, when it comes to A's and B's, we have the opposite antibody. Okay, so this has no A's and no B's. It's going to have both antibodies. Antibodies against A, like this triangle that would fit on a capital A, right? And antibodies against B. Would this person have antibodies against Rh? We already determined earlier that an Rh negative person would make them if they're exposed. Now, if this person possibly has, so I'm going to put a question mark there, right? These antibodies that would bind to the Rh factor. And now I ask this last question, who can they receive a transfusion from? And I ask, well, who will somebody who's O negative attack? They'll attack all A's, all B's, and possibly any cells with the Rh factor on it. We don't know for sure, but would you want to take that chance, right? Somebody has negative blood, I would not want to take that chance. So I can't give them A's like on A cells or on AB cells. I can't give them B's like on B cells or on, not B cells, you know what I mean, red blood cells with B's on them. And I can't give them anything with Rh on it because there could be antibodies against Rh in this person's bloodstream. All I can give them is O negative blood. Now, this person's kind of, really limited, right? But if we consider this person and ask the question, who can they donate to? Well, this couldn't be better blood for donating, right? There's no A's to attack, no B's to attack, and no Rh's to attack. An O negative person could only receive from another O negative person, but anybody else could receive this person's blood, right? This person could donate to anyone. Universal donor, right? All right. One more example with blood typing. We'll do a couple of genetics questions. Okay, let's do A negative blood. Okay, what antigens do they possess? Let's draw the picture, right? We have A negative. I'm going to draw a picture of a cell with an A on it, no B's, no RH's. So there you go. What antibodies do they possess? Okay, it's going to be the opposite when it comes to A's and B's, so I'm going to have antibodies against B. When it comes to RH, well, this person is negative. They may have been exposed, in which case they would have antibodies against RH. So I put a question mark. I'm not sure. Who can they receive a transfusion from? Well, I don't want to give them B's. And to be safe, just in case, I don't want to give them RH factors. So, no positive blood here. That's off limits, right? But I can give them anything that doesn't have a B otherwise, right? So I can give them A negative and O negative. So you can see how somebody who's got positive blood actually has more opportunity for transfused blood than someone who's negative. Okay, the last three slides, I want to talk about some kinds of genetic questions. And these are among some of the questions that I have up for practice questions to get you ready for the quiz. Okay? So let's take a genetics question like this. This is number 9 in your questions, so I'll do this one with you. Can an AB father with type AB blood, have a type O child? An AB father, type O child. Now, a lot of people, if I just ask you that, think about it. What do you think? Can an AB father have a type O child? Now, there's going to be a mother too, right? Don't forget that. What a lot of students will tell me, okay, maybe you've made up your mind, absolutely, if there was an O mother, this could be. But we have to look at this and understand that each parent has to give a gene to their child. So one way to approach this is we write down what we know. I know that there's an AB parent. So I write down AB is going to be crossed with another parent, right? There's going to be some genes from one parent, some genes from the other parent. And we don't know, question marks, right, what the other parent is. So let's say the mother, just for argument's sake, is type O. They're going to have 2 genes, right, for O. Okay, so one way to approach these kinds of genetic questions, just to prove it to yourself, we make a Punnett square. Put AB on one side and OO on the other side. I know some of you probably remember these, and maybe you don't love this stuff, but this is an easy way to prove it to yourself. AB parent. Mother is O, but they can only give one, right? I see not even one single child that is type O. So if this is the Maury show, right, or Springer or whatever show, and they're trying to figure out if this guy is the father, absolutely not, right? This would make a good show. Somebody's going to get slapped. So this is not the case, right? Let's do another one. Can an A mother and a B father have a type O child? This is similar to number 12 in your questions. A mother, B father, they come together, right? So we write down what we know. Let's get started. A cross with B, right? Now, most students, a lot of times, will tell me, well, they're all going to be AB children. No O children. Because you got an A parent and you got a B parent. But we have to think about what are the different ways you can be type A, okay? So let's write those down. There's more than one way, right? You could be AA or you could be AO, correct? Now I'm leaving off all of the I's here just to keep things simple, right? We know that we're talking about genes when we pair them up like this, right? So AA or AO. And for a B person, there's 2 ways as well. So what we're really talking about is many different possibilities between these. It could be the top 2 together, the bottom 2 together, or some other mixture. So we're really now talking about 4 possible crosses among 4 possibilities here. So we could do all those, right? AA and AB, and BB. All the children are going to be AB. So if you thought it was going to be AB, absolutely, there are going to be some of those in the mix. But then we have AA versus BO. And now I see a few, let's go with red here so you can see this. Now I see a few situations where the children are not AB. Now I do the next one with AO and BB. Now I see some that have type O blood, or sorry, type B blood, okay? And then this last possibility here, right, these between the bottom 2, I see an example of AB, BO, AO, OO. We could have a possibility for any blood type, though I'll admit the possibility of having a type O child with the information we have, right, and all these little squares, 16 of them across the way here. With what we know, there's only 1 chance in 16 little squares, right, that the child would be type O. So it can get a little more complex, right? You got to be able to think about this. Think about the fact you're going to get more. You're going to get 1 gene from one parent and another gene from the other parent. You can't get 2 from one and 0 from the other. Okay? Okay. 1 more, and then we're done. Okay? And this question is exactly questions 13 in your practice questions. Okay? What are the chances of an RH father and RH negative mother having an RH positive child? And then same question, but what are the chances of having the RH negative child? Okay, so now again, we write down what we know. One parent's positive, the other parent's negative. So here you go, right? RH positive crossed with RH negative. All right, so I have to now think a little bit. How many ways could you be positive? There's more than one, right? But there's only one way to be Rh negative. You'd have to have 2 recessive genes for it where you don't make the factor at all. So I'm going to use the letter D, right? Because it's another way of talking about the Rh factor. Okay, so capital D would be you make the Rh factor. Big D, right? But you only need 1 of those genes, right? So if you have 1 big D and then a little d, you'd still be Rh positive. And then you'd have to have 2 little d's together, right? 2 genes that didn't give you the Rh factor to be Rh negative. So we really have 2 possibilities here, and we don't have any other information to go on. So let's consider 2 big D's against 2 little d's, and then a big D and a little d with 2 little d's. So here we go. Big D's with little d's, and we get all big D, little d as a result. So in other words, every possible child here, the chances of having Rh positive, a big D, is 100%. But we got to do the other one. And when we do that, we see that it's 50-50, right? Now, let's take them all together and answer the question. When I count up the big D, little d possibilities, I got 1, 2, 3, 4, 5, 6, and there's 8 little squares all together, right? So 6 out of 8, or 3 out of 4, that's 75% Rh positives, right? and there's only 2 out of 8, or 1 out of 4, 25%, Rh negative. All right, and that is therefore your answer. Now, if you knew more information, right, you could actually maybe give me different answers, right? Like, what if I were to tell you that, let's see, the person who is Rh positive, okay, let's see, what are we going to do with this? The person who is Rh positive, I tell you that for sure they have 2 genes for the Rh factor, 2 big D genes. If I were to tell you that up front, I'd have to get rid of this as a possibility, right? And then you'd actually go and tell me 100%. Let's say we get rid of that. Let's say I give you a different piece of information. Let's say I tell you that this person who is Rh positive, his mom was O negative. All right? His mom had 2 little d's then, right? Had 2 genes, neither of which gave you the Rh factor. If I told you that this person had a parent who had a negative blood, you know that automatically there's a little d in the picture. And then we'd get rid of this one altogether, wouldn't we? And then I'd have to say it's 50-50, right? So it all really depends on what information you have. All right? Okay, I'm going to leave it at that, guys. This is the last bit I'm going to throw at you. Go and try those practice questions, see how you do. All of the answers to them are also posted.